Hello, everyone. Welcome to today's broadcast, Developing a Robust Critical Quality Attribute Monitoring Method via Multi-Attribute Monitoring Principles for Therapeutic Monoclonal Antibody Development, Manufacturing, and Lifecycle Management. I'm Rita Peters, Editorial Director of BioPharm International, and I'll be the moderator for today's event. We're pleased to bring you this webcast presented by LCGC and BioPharm International magazines and sponsored by Waters Corporation. I'd like to share this message from the sponsor. With 60 years of research and development of specialty measurement instrumentation, Waters' LC-MS, chemistries, informatics, and thermal analysis innovations help pharmaceutical and biopharmaceutical companies efficiently accelerate and improve workflows. The company's deep understanding of the global pharmaceutical industry, combined with its advanced analytical technologies, help bring critical medicines to patients quickly, safely, and effectively. I've got a few important announcements before we begin. The webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which is located on the right-hand side of your screen. You can enlarge the slide window by clicking on the small square icon in the upper right-hand corner of the slide window, or you can hover your mouse over the lower right-hand corner and drag the window to the desired size. The slides will advance automatically during the event. If you have any technical problems viewing or hearing the presentation, just click on the question mark Help widget in the dock at the bottom of the presentation window. Now I'd like to welcome today's speaker, Samantha Ippoliti. Samantha recently joined the Scientific Operations Biopharm Group at Waters Corporation from Novartis Institutes for Biomedical Research. During her time at Novartis, she worked as part of the Analytical Characterization Team for Biopharmaceutical Developability Assessment. Her areas of expertise are in LC-MS-based protein characterization, including intact subunit analysis and peptide mapping of monoclonal antibodies, antibody-drug conjugates, fusion proteins, and bispecific protein moieties. Since joining Waters, she has primarily focused on MS-compatible ion exchange chromatography and its application to biotherapeutic charge variant analysis. So thank you for being here today, Samantha. I'm going to turn the presentation over to you.
Thank you, Rita, and thank you to all of you for joining us today in our third installment of BioAccord-focused webinar series. If you missed any of the first two sessions, you can find these links in your Resources tab on your portal for this webinar. And today we'll be discussing developing a robust CQA monitoring method for therapeutic monoclonal antibody development, manufacturing, and lifecycle management. So we'll be talking today about critical quality attribute monitoring in the biopharm industry. First of all, what is a CQA and what are we going to be looking for those? Secondly, what factors are there for a successful implementation of a CQA or MAM approach for intact and subunit monitoring? Now, you've already heard about the BioAccord system in the first two webinars, but we'll take a look at how this system can help you in the successful implementation of some of these methods. Then we'll take a look at a few industry examples of subunit MAM on traditional high-res mass specs. And then we'll take a look at how we can use BioAccord to do this in a simpler way. And we'll look at a few interesting case studies where this could be applied. The biopharmaceutical industry has grown tremendously in the past decades. In 2016, 8 of the top 10 selling drugs were biologics. Given the growth potential, developers are facing increased competitive pressures from both innovators and biosimilar producers, and they are therefore looking for ways to be faster to the market, lower their costs, maximize profits, keep their supply secure, all of which while keeping patient safety at the forefront. In order to do this, analysis methods must be streamlined, and our CQA's critical quality attributes must be defined. Many of these are going to be monitored with LC-MS-based methods. LC-MS-based CQA monitoring methods can provide higher sensitivity, and selectivity compared to optical methods that are traditionally used in release testing. This will also be able to reduce the number of release assays that need to be performed, and will be able to enable rapid at or online testing. An effective characterization and robust monitoring of CQAs during product and process development are fundamental to biopharm development and the lifecycle management of a drug. I'm sure most of you have seen a slide like this before, but it's a nice starting point for us in discussing the importance of characterization of biotherapeutics. As you can see, they're going to be much more complex and heterogeneous than something like a small molecule drug. Now, each of these characteristics on the right can have an effect on the efficacy and safety of a drug, so it must be fully characterized and monitored during discovery, development, and manufacturing. Today we'll touch on just a few of these: oxidation, glycation, and glycosylation, but all of which are going to be very important in the development of a drug. Now let's take a look at this from the regulatory perspective. The FDA published this retrospective evaluation article back in 2017. Now it showed that 79 of 80 approved BLAs, 2000 to 2015, MS for protein and impurity characterization, and the number of attributes per BLA is only increasing over time, and especially more so as these drugs are becoming more and more complex. As you can see from the graph on the top right, got mostly antibodies at this point, but we're increasing to proteins, fusion proteins, antibody drug conjugates, and even more complex biological drugs coming out on the market today. LC-MS support is already heavily used in characterization, but will also increase in the areas shown here in a much broader scope, including structure function studies, process development, formulation development, regulatory support from IND to BLA, and then in post-approval comparability studies. This will be especially prominent in the QC and comparability as time goes forward. With all of this additional MS support, there will be need for more and more people who can support it, meaning MS will become much more widely accessible and easier for users to actually use. Now, in order to do this, we're trying to be able to expand the number of scientists who can generate high-quality MS data, ensure that the data can be acquired, analyzed, and reported in a compliance-ready manner, and this will enable faster decision-making and overall greater productivity. Now, you've already gotten a good introduction and overview of the BioAccord system in the previous webinars. This was launched back in January, and I won't be going too much detail about the system, but just a quick refresher to your memory. We've designed this new benchtop TOF system with customer-oriented workflows in mind. A little bit more on that on the next slide, but in general, the system itself consists of the Acuity I-Class with either a TUV or a fluorescence detector and the new RDA detector. All of these controlled by compliance-ready Unify software. Now, the I-Class will deliver robust separations, and the RDA offers high-quality MS data in such a small, compact TOF MS system. It also has a Smart MS feature, which is a self-diagnosing function offering step-by-step instructions if troubleshooting is ever needed. All right, now I mentioned workflows. The best part about this is we're working with a mass range of up to 7,000 mass to charge, making it ideal for biopharmaceutical analysis of the 4 workflows that you see here. Intact mass on the high end, obviously, and since it's up to 7,000 M/Z, we're able to look even at native analysis. We can look at something as small as a release glycan with the fluorescence detector and have the mass confirmation as well, and then even down to peptide mapping and monitoring. Now, you've probably heard of monitoring most commonly with peptide mapping, but today we're going to focus and dive in a little bit more in the subunit and intact realm. Now we know that CQAs are important, and now we have tools to characterize and analyze them, but how do we go about doing it in the most efficient way possible? You've probably heard of the term MAM, multiple attribute monitoring, which represents a recent trend invested by many pharmaceutical companies improve productivity and data quality by effectively and directly monitoring multiple CQAs simultaneously within just one MS method, LC-MS method. This methodology has been evaluated in biopharmaceutical production and bioprocess development, aiming for PQA screening, process development, and optimization, including formulation development, etc., with the potential to ease method transfer and filing as well as adapt to future practices, such as online or at-line, near real-time product release and continuous manufacturing. During this evaluation exercise, the biopharmaceutical industry has experienced the excitement as well as some challenges of deploying MAM, especially into the regulated environment. Thus, many deployment considerations need to be made in the early phases of looking into MAM as an option for the organization. MAM methods, they need to be based on thorough understanding of the product through an in-depth characterization. You need to look at different compliance expectations when applied to development and manufacturing and quality control. Methods need to be easy to use, robust, and automated to provide operational efficiency. This is where we can put our BioAccord system. And since the method is intended to be applied to laboratories traditionally not familiar with MS-based assays, some resource investments such as training, time, and cost need to be considered. So most commonly, you've probably heard this term going towards a peptide mapping assay. And I mentioned today we're going to be looking at the subunit and intact level. So let's take a look at how some people are already doing this. Now I'll talk about a few examples of, again, what's already happening out there using subunit MAM-based methods with the use of high-resolution QTOF systems. This technology is proven, as shown in this peer-reviewed article from Janssen, that was used to support cell culture modulation. The workflow can be automated to provide high-throughput, sensitive, and reproducible multi-attribute monitoring results. This example in particular, they were looking at glycosylation at some of their monoclonal antibodies. And the method itself, again, is going to be automatable, and it produces highly reproducible results for the relative glycoform quantitation, supporting their DOE studies. In this case, they were also looking at aglycosylation and afucosylation so that they can adapt their cell culture conditions if they needed to. The second example from Janssen shows a subunit MAM approach to monitoring oxidation levels in various MAB candidates that they were looking at. The first panel, Panel A, shows the sample prep workflow in a little bit more detail, NDOS and IDES digestion, followed by reduction, a total of 45 minutes of incubation time. Now compare this to a regular peptide mapping analysis where you're spending half a day or overnight protocols preparing your samples. This cuts down quite a bit of time. Panel B, it will be showing the data workflow from acquisition, report generation, and as you notice, this is also much shorter than a typical peptide mapping method. This one is under 10 minutes of separation, and you can still see the 3 subunits that you're expecting to see. Now, if you look to the bottom right of this slide, see that they report about 2% oxidation for this particular MAB, and they performed a variety of different variability testing for anything from columns, analysts, day-to-day variability, all giving in the 2 to 2.1% oxidation, and all of which have under 10% RSD. So you can see that's a very reproducible method that they're able to apply to many different MABs, and they can use it as some of a platform method. These are both great examples of how subunit MAM can simplify method development and increase throughput, and then therefore enable faster decision making. But they are using high-end QTOF systems that typically require MS experts with lots of experience and training. Now let's take a look at some of the work we've been doing in-house with our BioAccord system. To start off our experiment with an intact and subunit CQA monitoring approach on BioAccord, we first wanted to test out the basics. So we took a simple case of IDES digested and reduced trastuzumab And we're looking at light chain and FD glycation, as well as the N-glycosylation profile on the SC as our CQAs. Now we performed triplicate injections of the digest on 3 different columns with different lots, and repeated the whole experiment on a second BioAccord system. Method details are shown down here on the right. Very typical reverse phase conditions, all in a 15-minute method. Now for all of the subunit data that you're going to see today, it's all done in this exact same LCMS method. So it's going to be somewhat of a platform. Now notice the RDA setting. There's only a few to set, much less complex than a typical high-res mass spec method, and that's just the way it was designed, to be much more accessible to the users. Now one important part of any AM-based approach is chromatographic reproducibility for the retention time. We're doing this in any sort of compliant, automated fashion. Retention time must match up in order for peaks to be correctly identified. Notice the chromatogram in the top right of this slide, and that's exactly what we have for this particular triplicate injection that I'm showing. As I mentioned, we're going to be looking at the light chain, FD, and FC CQAs, glycation and glycosylation levels, in addition to the identity of all these species. Each of the peaks shown here has been positively matched by both retention time and observed mass within 20 ppm set threshold in the method, and once identified, is calculated in relative to the parent species and can be reported. All of this is automated in Unify software from acquisition, deconvolution, peak assignment, calculation, and reporting. Let's take a look at the reproducibility. First, we'll look at the glycations, low-level species right here. First, we focus on the glycation of the light chain on the left of the slide. Highlighted in blue is going to be the first system, and in orange is going to be on the second system. And in the blue box will be the first column, orange will be the second column, and red is the third column. Have very good agreement from each of the 2 systems in 3 columns. For the light chain, we get 1.6 to 1.8% glycation as a result, with only 6.5% RSD over all of the injections. For the FD, on the right side of this slide, same color scheme as we're looking at. System 1 is in blue, and System 2 is in orange, each of those 3 columns. And for the FD glycation, we have 1.1 to 1.3% glycation and 7.9% RSD over all of the injections. So even at this very low level of modification, we still get very reproducible and repeatable results. Now we take a look at the N-glycosylation for trastuzumab. And again, you can see very reproducible results across all the systems and columns. As you can see, now I've gone to the low levels. You can see it's very, very reproducible, even at the low level. For species that are above 0.5% relative abundance, the percent RSD is less than 10, and most of these are even below 5. The results shown here are also very comparable to released N-glycan results that have been formed for other batches of trastuzumab in our lab previously. And similar to the Janssen example from before, this N-glyco profile can be monitored during process development or expression screening to obtain a desired profile and be able to make fast decisions and adjust along the way if needed. Going along that same trend, the afucosylation levels in this example could also be monitored, and in this case, they're contributing up to about 12% of the total glycoprofile. For our second case study, an example of CQA monitoring, chosen sequence variants. Now, amino acid sequence variation is caused by either genetic mutations in cell clones or misincorporation of amino acid cell culture. So this is a CQA that's typically monitored in expression screening and process development, ensure that we're really getting the product sequence that we're aiming for. In our case, we're using a sequence variant sample of trastuzumab. It has 1 point mutation in the light chain, causing a plus 14 Dalton shift, and 2 point mutations in the heavy chain, more specifically in the Fc, which results in subtracting 32 Dalton from the heavy chain. Now, if you're doing the mental math there, that means minus 36 Dalton for the full antibody. It's a small enough shift that could make intact analysis pretty difficult, especially if there's multiple forms of the variant present. So for this, we're going to subunit approach using the exact same sample prep and LCMS method that you saw previously in our previous example, except for this one, we're going to include a deglycosylation step to simplify the data processing and data analysis. Sequence variant sample, in this case, was spiked into the originator in a serial dilution to resemble a typical working range that we would be interested in seeing for a sequence variant example. Now on this slide, you can see some of the data that came out of this example. On the top of this, you see the 3 very well separated subunit species, Fc, L light chain, and Fd. And below, you can see the assigned peaks correctly assigned by Unify after deconvolution. The sequence variants are correctly identified, and then Unify will also calculate those out for us in relation to the parent. As you can see on the right side, plotted the linearity of the serial dilution results, and we have nice linearity and reproducibility in this range from about 1 to 50% variant. For our third case study, this one is one of my favorites, actually, cetuximab. It's an interesting example of an antibody that has 2 different glycosylation sites, and it's going to be one that a subunit level analysis will be directly benefiting us in comparison to some of the other methods out there. So this antibody has not only the standard Fc glycosylation, but another site in the Fd. Again, you also know as the heavy chain. So that's going to be 4 sites in total in the intact full antibody. So if we were to look at release glycan assays, which is the standard for glycoprofiling, we would only get the global profile, not knowing which glycans belong to which site. Now that can be important because each of these domains of the antibody have a different role to play biologically. The same is true if we were to look at a simple reduced light chain heavy chain analysis. So the Hyde's digestion and subunit approach allows us to investigate each of these domains separately and obtain unique glycoprofiles for each one. Now here you can see the results from our subunit analysis of cetuximab. Distinct profiles for both the Fc and the Fd. Again, these are assigned within 20 ppm accuracy, which we set in the method. The Fc shows a typical N-glyco pattern for IgG1 Fcs, while the Fd gives quite a different range of species. The most dominant ones, including some of those alpha-galactose and non-human sialic acid species, which used to doing normal antibodies, something you might not be used to seeing. And also we see a mix of bi- and tri-antennary structures. These findings are also in agreement with previously published results, so we're in good confidence that we're looking at what we're supposed to be seeing. Let's take a deeper look at some of the relative percentages of each of these. Here we see the Fc glycosylation on the left side and the Fd on the right side. This was a triplicate injection experiment, and as you can see, or maybe not, the error bars for this analysis are almost non-existent, meaning we really have very reproducible results across this analysis. Percent RSD for each of these is going to be less than 3%. And as you can see, even some of those low-level species still have great reproducibility. This is just one example where a subunit-based analysis gives us a distinct advantage over the standard release glycan assay, and we can do this quickly, easily, and reproducibly with BioAccord. As I mentioned before, and now again, the 3 examples discussed so far have all been run on the same LCMS gradient and system, suggesting this has true platform generic method capabilities. Our last example of an intact-based CQA monitoring application is ADC DAR analysis. Now this will be a full intact antibody, unlike the subunits you've seen before. DAR, or drug to antibody ratio, specific CQA for ADC biotherapeutics, which can be influenced by conjugation processing, formulation, storage, etc. So therefore, it must be monitored throughout its life cycle. For this analysis, we'll perform intact LCMS. We want a total overall DAR result for the antibody. The deconvoluted spectrum to the bottom left of this slide shows drug distributions from 0 to 8 drugs, and the calculated value for this will be about 3.5. As you can see in the screenshot to the right, this was all done with Unify, which has automated peak detection and DAR calculations, making it much more convenient for screening and process development. So then we can take this also to a comparability assessment. You can see in the mirror plot, we're looking at an innovator on the top and a biosimilar on the bottom. You can see the deconvoluted mass spectrum here. Again, not deglycosylated, so it is complex. And see the differences immediately from the mirror plot. You're losing some of the higher DAR species and biosimilar. So as a result, our DAR for the innovator will be 3.5, and the biosimilar is going to be 3.15. Something you can easily pick up differences quickly. Slide will show you just a little bit more detail about the ADC innovator and biosimilar example. On the left side of this slide, we're looking at the reproducibility of these, again, on 2 different systems of BioAccord. System 1 is shown on the left, System 2 is shown on the right, and the innovator will be in blue, and the biosimilar is in orange. As you can see, the very similar results on the 2 different systems. The table on the right here shows a little bit further detail of the triplicate injections that were done, and the standard deviation for the DAR results was less than 0.1. Great to see. These examples will show that take a decently complex analysis and make it pretty routine. In summary, I hope that I've convinced you today that MAM-based approaches to CQA monitoring are becoming increasingly adopted in the biopharm industry for characterization, process development, and QC release. Also, that increased MS accessibility and ease of use are crucial to the successful implementation of MAM-based workflows. And lastly, that the BioAccord system enables fast, robust platform methods for intact and subunit-based CQA monitoring with the benefits of simple sample preparation, automated data analysis, and all of this in a compliance-ready environment. With that, I'd like to thank the following team members, and of course, thank you to you all for listening in. I believe at this point we should have some time for questions.
Well, thank you, Samantha, for the presentation. Before we move on to Q&A, just a quick reminder to the audience that they can submit questions by typing them in the Q&A box, which is located on the right-hand side of the presentation window. So let's go on with the questions. Here's the first one. Can you comment on the benefit of monitoring quality attributes at the subunit level compared to the peptide level?
Yes, of course. As we've seen throughout this presentation, I think that one of the biggest things is that it's much simpler and faster doing this at a subunit level. You're also getting some less artifacts due to the shorter sample prep time, also shorter runtime, and definitely simpler result interpretation. So with a peptide map, let's say you have tons of peaks that you're looking at and identifying. With this, you're only looking at really 3 chromatographic peaks and seeing what's under those. Many of our customers have found it very attractive, especially for those high throughput analyses, meeting turnaround times, such as in bioprocessing support. Thanks for that one.
Okay, here's another one. For the sequence variant analysis, what was the lowest level you could detect?
So for that one, I would point out quickly that we're really looking at co-eluting species here. So for that, we can see down to about 1%. And that's generally a good area to be at when we're looking at impurities at this level. We do know that some other published results show that if you extend the gradient quite a bit, and you can actually separate things out chromatographically, you may be able to lower that LOD. Some of the published results, if I'm remembering right, were about 0.3%.
Okay, thank you. Here's the next one. So if you see a mass change at subunit level, how sure are you for the correct modification assignment, such as how to differentiate trisulfide bond versus double oxidation, since they are both plus 32 Dalton addition?
So actually, that's a good question. Before we're deploying any of these subunit monitoring methods, we're always going to characterize the molecule at all levels. So we know exactly what we're looking for and what we're monitoring for. So we'll look at the intact and subunit as well as peptide mapping to really confirm what we're seeing at the subunit level is actually true and is actually there.
Okay, thanks. Here's the next question. Would this be able to monitor deamidation?
That's a great question, actually. Very related to the last one. So when we look at things like deamidation, at the subunit level, we're really not getting down to the 1 Dalton difference. So for that, we would actually need to go to the peptide level. And even when you're down at peptide level, they can still be complex even if you don't have that chromatographic separation. So yeah, deamidation is going to be a difficult one. Isomerization is the same thing. That's most likely going to be a peptide map method. Also for deamidation, you can look at, let's say, an ion exchange method will help you with deamidation. Thanks for that one.
Thank you. Yeah, here's another one for you. So regarding mass resolution, what kind of modifications could you see from this analysis, and are there limits?
Yep, so that one is very much related to the last one. At the subunit level, we're typically looking at things down in the range of oxidations, plus 16 Dalton. You saw from the sequence variant analysis that plus 14 was definitely detectable. But like I said, anything such as the isobaric or near isobaric species, we're going to need a an orthogonal method such as peptide map or ion exchange if that will get it for us. Thanks. Okay,
yeah. Here's another one for you. So what is meant by 3 replicate? Is that 3 injections from the same HPLC vial, or is it 3 sample preparations?
And I believe that probably means the trastuzumab analysis, I'm thinking. For that one, it just was 3 from the same vial.
Okay, thanks.
But definitely important to look at different sample preps. I didn't include it in this presentation, but we do have data that will show that it's about the same if you do it in multiple sample preps.
Okay, great. Here's another one for you. If I develop a subunit assay for the critical quality attribute and want to transfer to the QC lab, can the system meet the compliance requirements and be deployed into a QC lab?
Yes, absolutely. That's one thing that the system and these types of methods will shine at, actually. The BioAccord is operated under 21 CFR Part 11 and Annex 11 compliance. It's got ready informatics with Unify works in either a workstation or a network configuration. It does provide that comprehensive and searchable audit trail as well. If anyone's looking for more detail on that, I can go into it, but I think that covers that question.
Okay, great. Well, then we have another one for you. Does the system have new peak detection feature?
So we're actually looking at, well, if that means chromatographic or MS new peak detection. Unify does have a feature that has a comparison mode so that you can easily see new peaks as they come up.
Okay. Let's see, we have one more here. So can I perform subunit analysis and release glycans on the same system?
Currently, you would be able to. The BioAccord is actually sold as a total system solution. It originally comes with either the TUV detector or the fluorescence detector. So if you're interested in doing both of the workflows in one system, we're also offering a fluorescence and glycan add-on kit. So this will give you the fluorescence detector plus the additional startup kit with glycan applications, the libraries for Unify, and all that comes with it. This will all be configurable in a stack, and you can choose which of the detectors that you want to use at that certain moment, and you wouldn't have to shift around your detectors at all, so they can all fit together.
All right, well, thank you very much, Samantha, for your presentation and for taking the audience questions. I want to thank our audience members for attending and participating, and I'd like to thank Waters Corporation, our sponsor, for making today's webcast possible. Just a quick reminder, you can register and watch the entire 3-part series, and you can register directly on your screen to do that. I also invite you to take a 1-question survey that will pop on your screen at the end of today's webcast. You'll receive an email alert letting you know when this webcast will be available for replay. I invite you to forward that announcement to your colleagues who may have missed today's live event. So thank you again for joining us, and have a good day.